Understanding intravenous fluids requires some understanding of the basic physiology of fluid. In this initial vodcast, we will review the basic physiology applied to fluids in intensive care. This includes some important definitions, the concepts of water balance and sodium balance, body fluid compartments, and Starling's forces. Osmolality is the concentration of osmotically active particles per set weight of fluid, usually described as milliosmoles per kilo in biology. Osmolarity is a related term defined as the concentration of osmotically active particles per set volume of fluid. The former is usually the preferred measure because volume of fluid changes with temperature. Osmotically active particles may or may not be able to pass across a semi-permeable membrane. If they can, then they do not have the ability to generate an osmotic pressure, as the particles equilibrate in the available fluid space. Tonicity is a related and important property of fluids. When osmotically active particles cannot pass across a semi-permeable membrane, they are then able to generate an osmotic effect on the fluid. Tonicity, therefore, effectively determines the partitioning of water between the compartments, such as the intra- and extracellular fluid spaces. The normal osmolality of extracellular fluid is 270 to 290 milliosmoles per kilo. Consider, for example, the effects of a 350 milliosmoles per kilo hyperosmolar solution of sodium chloride. Sodium chloride is able to distribute across the extracellular fluid space, but does not pass across cell membranes. This will result in an increase in the tonicity of the extracellular fluid compared with the intracellular fluid, drawing fluids from the ICF to the ECF. This fluid is therefore regarded as hypertonic. This contrasts with the effect of a 350 milliosmolar per kilo hyperosmolar solution of urea, which can distribute across cell membranes. Because the osmotically active particles are distributed across the total body water, there is no shift of water as the increase in osmolality is shared across both fluid compartments. Therefore, even though this solution is equal in osmolality to the sodium chloride solution, it is not hypertonic. Both of these fluids have the same osmolality, but only the saline solution has a high tonicity. The tonicity of a resuscitation fluid is important as it has significant effects on how the fluid will equilibrate. Hypotonic fluids will result in the movement of fluid into the intracellular space, causing cells to swell. Sudden significant changes in plasma tonicity results in a potentially significant shift of water, which can be clinically relevant. Examples include osmotic demyelination of the central venous tissue and exacerbations of cerebral edema. It is also important to red blood cells. If hypotonic fluids are mixed with red blood cells, the cells absorb fluid rapidly and the swelling results in cell lysis or hemolysis. Osmotic pressure is the theoretical pressure required to prevent net diffusion of water through a semi-permeable membrane with differing osmolalities of solution on either side. In other words, it is the hydraulic pressure that would oppose osmosis. When referring to the circumstances where proteins are generating the osmotic pressure, the term oncotic pressure is used. The normal tonicity of the blood is 270 to 290 milliosmoles per kilo. Fluids with a similar tonicity are known as isotonic with plasma. Osmolality can be directly measured using the freezing point depression test. A change in osmolality of 1 osmol per kilo depresses the freezing point by 1.86 degrees Celsius. Fluids are then measured relative to this benchmark. Fluids with a lower tonicity are known to be hypotonic, while those with a higher tonicity are hypertonic. Osmolarity is often estimated from the following equation. This is because these substances exert the greatest proportion of the osmotically active agents in the serum. It is clear from this calculation that the osmolality of the extracellular fluid is primarily determined by sodium salts. Hyperosmolality due to excess solute will have varying clinical effects dependent on whether the solute distributes across the total body water, such as alcohol, if it is contained in the extracellular fluid, such as hypertonic saline, or if it is retained in the circulation, for example concentrated albumin. In circulation, the plasma colloid oncotic pressure is around 12 mm of mercury, while the surrounding interstitium has a colloid osmotic pressure of around 8 mm of mercury. This is largely attributable to the presence of albumin both on the plasma and in the interstitial space. The average total water content, known as total body water, is often measured as a percentage of total body weight. In males, water constitutes 60% of body mass, while in females it is 50%. This percentage decreases with age as muscle mass falls, fat content increases, and connective tissue and bone constitute a greater proportion of the total body mass. Water distributes throughout a theoretical space known as the total body water. In itself, it is not restricted by any membrane or boundary and moves along osmotic gradients. Traditionally, total body water is compartmentalized into the intracellular fluid and the extracellular fluid. Extracellular fluid is approximately 1/3 of all body water and is composed of plasma, accounting for approximately 1/4 of the ECF, and the interstitial fluid. The principal cation and osmotically active agent in the ECF is sodium, and as such, ECF volume is largely determined by the regulation of sodium balance. ICF generally constitutes 2/3 of all body water. Intracellular fluid is composed of water within cells, and unlike ECF volume, is principally regulated by overall water balance, which is itself regulated by hypothalamic osmoreceptors. The three major compartments, intracellular space, interstitial space, and the intravascular space, are separated by water-permeable membranes, with the result being that they contain almost identical concentrations of osmotically active particles. In short, total osmolality of the body determines the total body water. As osmolality rises, total body water is increased, and this distributes across the total body water space along osmotic gradients. The proportion of this that is in the extracellular space is determined by total sodium balance. Maintaining hydration is an essential homeostatic mechanism of the body and is determined by the balance between intake and water loss. Intake of water averages 2.5 litres per day and is comprised of oral fluids and that contained in food. It also includes water produced as a result of metabolic activities. Water balance is achieved if losses exactly match intake. Water can be lost from the skin, in the faeces and urine in assisting to clear waste, and through insensible losses, such as through respiration. Control of water balance, therefore, involves controlling both input and excretion. The proximal tubule, loop of Henle, and distal tubule of the kidney retain almost 95% of filtered water and sodium. Regulation of water balance is largely controlled by the handling of the remaining 5% as it passes through the distal nephron and collecting duct. As this water continues through the distal nephron, it passes through the medulla of the kidney. The interstitium of the renal medulla is incredibly hypertonic, a result of the countercurrent mechanism of the loop of Henle. In the absence of antidiuretic hormone, these tubules and ducts prevent reabsorption of water, and the remaining urine is excreted in a very dilute form. At maximal diluteness, the urine osmolality can be around 50 milliosmoles per kilo, significantly less than the average plasma osmolality at 280 milliosmoles per kilo. Through this mechanism, excess free water can be excreted from the body. In contrast, in the presence of antidiuretic hormone, these tubules become very porous to water, which is then reabsorbed by osmosis due to the high osmolality of the medullary interstitium. In this way, free water can be retained, and at its maximum, urine osmolality can approach 1,200 milliosmoles per kilo. Antidiuretic hormone increases water permeability by stimulating the production of aquaporin. This water channel appears on the luminal surface of the principal cells in the distal collecting system of the nephron as it runs through the renal medulla, allowing the retention of water. The degree of ADH secretion determines how much water is retained. Without ADH, water reabsorption is almost zero in the medulla. A water diuresis ensues. In the presence of ADH, most of the residual water is reabsorbed, resulting in maximal concentration of the urine. ADH also increases the uptake of urea by the collecting ducts, which further increases the medullary gradient. It also has a permissive effect on blood pressure. It is required to enable some vascular reactivity, allowing maintenance of normal blood pressures. It is sometimes, therefore, known by its other name, arginine vasopressin. Hypothalamic neurons, whose axons terminate in the posterior pituitary, release ADH directly into the blood. A number of mechanisms stimulate the release of ADH. In low blood pressure states, cardiopulmonary and arterial baroreceptors detect the low pressure, resulting in the decreased stimulation of the hypothalamus. This results in ADH release, increasing water reabsorption, with the biological intent of restoring intravascular volume. As previously discussed, vasopressin also has direct pressure effect, particularly when released in larger quantities. ADH release is inhibited by normal baroreceptor tone. Osmoreceptors detect a change in the osmolality of the extracellular fluid in the hypothalamus. A rise in the osmolality stimulates ADH secretion, resulting in the retention of water from the kidney, thereby reducing body osmolality. Conversely, a drop in the osmolality inhibits ADH secretion, allowing the clearance of free water by the kidneys. Normally, if osmoreceptor and baroreceptor stimuli oppose each other, the osmoreceptors will override, meaning that osmoreceptors can respond to a 1% change in baseline variation, whereas baroreceptors only respond to larger changes. However, larger drops in blood pressure in spite of a low osmolality will cause volume reabsorption to take precedence over osmolality. This has clinical relevance in the hyponatremia associated with low flow states, such as in chronic heart failure. Thirst is a behavioural mechanism that increases free water intake. Centres that mediate thirst are located in the hypothalamus and are powerfully stimulated by volume depletion and increased osmolality. The normal concentration of sodium in the extracellular fluid is tightly conserved. Normal values range from 135 to 145 millimoles per litre, representing the vast majority of extracellular cations. The proximal nephron controls reabsorption of the vast majority of water and sodium in parallel. Independent control of sodium and water takes place in the distal nephron. The major controllers of sodium handling are aldosterone and the natriuretic peptides. Aldosterone is a mineralocorticoid produced by the adrenal cortex. Its primary function is to increase sodium reabsorption in distal tubules and cortical collecting ducts. The most important controlling factor is circulating levels of angiotensin II, and this in turn is controlled by the release of renin in response to stimuli such as low glomerular filtration. The body interprets this as a sign of relative loss of extracellular fluid. The additional sodium that is retained distributes across the extracellular fluid space, increasing its tonicity. This stimulates the release of ADH, which increases the retention of water, effectively expanding the extracellular fluid space. In the presence of maximum stimulation by aldosterone, up to 2% additional filtered sodium is retained, accounting for almost 30 grams per day of sodium chloride. This seemingly small amount significantly increases total sodium stores. Aldosterone also acts on other organs to increase sodium retention. Natriuretic peptides, ANP and BNP, are secreted by atrial myocardium and to a lesser extent by the ventricles. These peptides are released by stretch of myocardium, a marker of overfilling of the vascular space. Both peptides stimulate the kidneys to increase sodium excretion by dilation of the afferent arterioles and increased glomerular filtration rate, and by inhibition of sodium reabsorption in the collecting ducts. This is the primary mechanism by which the body regulates the volume of the extracellular fluid. As discussed, the main function of antidiuretic hormone is the increased reabsorption of water, thereby diluting its concentration rather than reducing total sodium itself. However, it does increase sodium reabsorption by cortical collecting ducts to a small degree. A number of other hormones have minor effects on sodium balance, as shown here. Salt hunger is another behavioural adaptive mechanism that arises in salt deficiency. Ernest Starling's concept of fluid movement at the capillary level dates back to the late 1890s. He proposed a model as shown in the diagram, where the balance of hydrostatic and osmotic forces applied at the capillary resulted initially in the outward movement of fluid, and then later the inward movement of fluid into the capillary. At the start of the capillary, the outward forces predominate, allowing fluid to move into the interstitial space, carrying with it important nutrients. By the end of the capillary, the forces were mostly reversed, resulting in movement of fluid back into the vascular tree. A small amount of fluid remained, which he proposed was returned to the circulation via the lymphatics. Starling's forces are seen as a dynamic process, with the values changing across the length of the capillary, such that by the end the net movement of fluid is inwards. Hydraulic conductivity is effectively a constant describing the permeability of the vascular endothelium, which is specific to the type of body tissue. The blood-brain barrier provides a classic example of a poorly permeable membrane. While some of the basic assumptions of Starling's work have been challenged, the basic premise remains relevant. The key factors in the flux of fluid in and out of the interstitial space relate to the hydrostatic and oncotic forces. Importantly though, it has been increasingly recognised that this model changes during pathology, particularly with respect to the permeability of the endothelial layer. As previously described, Starling's forces change across the length of the capillary, resulting in a reversal of flow from inward to outward by the end of the capillary. This results in the removal of almost all of the filtered interstitial fluid. However, some remains and is returned to the circulation via the lymphatics. In this video, we have reviewed some important fluid-related concepts. The total body water is a theoretical volume through which water freely distributes. Conceptually, it comprises the extracellular and intracellular space, representing 1 and 2/3 of total body water respectively. The former is further divided into the vascular tree, which is almost 25% of the ECF, and the interstitial space. The movement of osmotically active particles that exist in the total body water may be restricted by semi-permeable membranes. This can affect the movement of water within these fluid compartments, a concept known as tonicity. The volumes and constitutions of these various body fluid compartments are largely regulated by two intertwined processes, sodium and water balance. Water balance is largely determined by the action of antidiuretic hormone, which increases permeability of the distal nephron to water, resulting in water absorption and maximal retention of water. Parallel behavioural mechanisms such as thirst increase water intake. Sodium balance is largely determined by renin-angiotensin-aldosterone axis and the counterbalancing natriuretic peptide mechanism. In short, water balance regulates osmolality, while sodium balance controls the volume of the ECF. Finally, Starling's forces demonstrate the importance of osmotic and hydrostatic forces on the essential flux of fluid between the intravascular and interstitial spaces. In subsequent modules, we will review how specific fluids influence and are influenced by these physiological concepts.